Hello, my name is Erica Fernandez, and I'm working with Evan Bate on determining the physical fate of microplastics within an aquatic ecosystem. Plastic has been the foundation of our modern-day society for years, and it is often used in our daily life, with 8,300 million metric tons being produced as of 2017. The production of plastics has led it being used in many of our key fields, such as the medical, food, and manufacturing industry. But of that usage, only 30% are in use, with the other 60% being discarded and 10% being recycled. Due to their lightweight as well as their mass production, they can cause issues for waste management as they easily travel into waterways via wind, municipal waste, or just through littering. Many of these plastics appear to persist on the surface waters, but unfortunately that's not the whole picture. Plastics are unique compounds with diverse characteristics and traits. And this especially applies in the way in which they biodegrade or float, as the amounts on the surface do not wholly represent the amount within the water column. This becomes a pressing issue as plastics are going into water sources such as the Great Lakes, with it being the largest surface freshwater ecosystem globally, as well as a major source of drinking water. As plastics are transported into our waterways, many aquatic organisms are impacted by it due to the constant contact as well as easily consuming it. Through this consumption, it bioaccumulates throughout the ecosystem as it progresses throughout the food chain. This is not only a physical hazard, but a chemical hazard due to their toxicological effects on species. Research has shown that the consumption of microplastics can lead to detrimental growth effects as well as reproductive effects on aquatic organisms. This can lead to fears about the impacts it will have on humans as these organisms are then consumed by us. With all the effects in mind, as well as their production, it's important to think about where they eventually end up when in our waterways. So our goal is to look into finding the physical fate of these plastics and their vertical distribution throughout a water column. One of our main hypothesis questions would be regarding the effects of density in regards to vertical distribution. Due to plastics having different characteristics and traits, many plastics sink very differently due to difference in buoyancy. However, one of our hypotheses is going to be looking at how biofouling will impact this as sinkages occur of lighter plastics. However, biofouling is not the only driver in plastic sedimentation. Plastic sedimentation in real-life scenarios can occur through many circumstances. Marine and lake snow is one of these cases in which plastic sticks to organic detritus and starts sinking towards the seafloor. This makes up around 70% of the lake snow that is seen within waters. A way that's similar to that is through it just sticking to sediment such as clay, adding density to these plastics. One of the more organic ways in which they can sink is through just being consumed by aquatic life and being digested as fecal pellets, further adding to the density. The main driver we're looking at in our project is biofouling due to the closed nature of our experiment. Biofouling is the growth of organic material on these plastics, and this can lead to changes in our buoyancy. The plastics that we're planning on using in this experiment are 3 of which that are different in their characteristics and buoyancy, and you have most likely come into contact with them within your daily life. The first one that we're going to be looking into is low-density polyethylene, otherwise known as LDPE. This is seen in thin films such as grocery bags, garbage bags, and whatever thin lining you can think of. This is expected to be the last one to leave the surface of our mesocosm due to the light nature of it. The next of these plastics is polystyrene, otherwise known as PS. This is commonly used and installed into many of our appliances and food packaging due to it being able to insulate heat very well. This is very neutrally buoyant due to it being close to the density of water. This can lead us to believe that it is more sensitive to change from biofouling and will sink as time goes on. The next of these plastics is polyethylene terephthalate, otherwise known as PET. You can see this in plastic bottling and food packaging, and this is the heaviest of the plastics. This is also used in many of our textiles, in our clothing, and our bedding. The area that we're going to be conducting this experiment on is the Tate Preserve. This is a former sand quarry found in Penfield, New York, and within this new formed lake, it is home to various fish species and benthic invertebrates. Before running the experiment, we wanted to see what was actually in the lake through running a blank water sample and filtering it through and examining it through a microscope. Through that analysis, we found that algae and daphnia were prevalent in the waters.
Liminal corrals are used for large-scale mesocosm projects, allowing for researchers to control an experiment that is subject to the natural environment. The purpose of a liminal corral-based study is to emulate the environment, thus making the results more credible and providing clear implications. With respect to the design of the corral, it's a cylindrical tube, 2 meters in diameter and 7 meters in depth. There's a central column that extends down the corral, and inside the column are various sampling tubes that capture water from inside of the corral at a given depth. So samples are taken at 0.5, 1, 1.5, 2, 3, 4, 5, and 6 meters. Also in the corral is a chain of hobo lockers, which are devices used to record light and temperature. So they also extend down to the bottom and are spaced 0.5 meter apart. And then sediment traps were also constructed and deployed to catch any particles that sank to the bottom of the corral. This could be plastic, any miscellaneous particles would all eventually enter the sediment trap. A peristaltic pump was attached to the end of each sampling tube to transport water in and out of the corral. Distilled water was rinsed through the tubing corresponding to each depth to mitigate any kind of buildup, so algal, sediment, etc. And then at each depth, 1 liter of the water sample was transferred into a 1 liter bottle. Surface samples were acquired by placing polycarbonate tubing into the water and placing a stopper underneath to capture the sample. The location of where in the corral to take the surface samples was determined by a quadrant system, and so we numbered each section of the corral and alternated quadrants each time samples were collected. And each time we took a surface sample, we would take two. We would have 2 quadrants selected each time. And the samples were taken frequently at the start of the study, and this was due to the fact that when the plastics were introduced to the lake water, they had not settled completely. But as this became more stable, samples were taken less frequently. A vacuum pump and an 8-micron gridded filter paper were used to filter the samples. So we found it easier to separate the plastic particles by size because then they'd be easier to count under the microscope. But I think most importantly, stratifying plastics by size gives us that ability to look at the relationship between the particle size and the vertical distribution. And so we used a 212 and 500 micron sieve. Both sieves were utilized for separation. And then each sieve was rinsed, and their contents were placed onto a petri dish to be counted. So when we were counting the plastics, we'd count from left to right, and then we would zigzag down. And when we first began, we would count together up until we could both differentiate between the plastics placed into the corral and miscellaneous particles, biofilm, daphnia, other microorganisms, etc. And then given the fact that each of the 3 polymers were assigned a distinct color, they could each be identified, and then they could be tallied.
Initially in the experiment, we conducted a lot of experiments at high frequency, being twice a week. This has led to an accumulation of a large amount of sample bottles that we weren't able to fully process and get results out of. However, what we can provide is a 1-week analysis of the plastic distribution. Although with only a week's worth of data, we can already see some discrepancies due to a very large amount of polystyrene at 6 meters, only 2 days after dosing. This can be viewed as a discrepancy simply due to the fact that it was the only plastic with amounts up to 400. This may have been caused by a large storm event that has happened the day before, and we are predicting this is due to the waves pushing against the liminal corral walls and causing mixing within the column. Other than that data, though, the LDPE appeared to follow the projections of having high concentrations at the surface and slowly decreasing as you get closer to 6 meters. With the PET, it seems to be concentrating towards the middle at 3 meters. 4 days after dosing, however, the LDPE follows the same trends of concentrating towards the surface, but polystyrene and PET concentrating towards the lower depths. But for the last day of dosing for that week, it seems that all plastics have a great concentration towards the surface at 1 and 0.5 meters. But LDPE is still following the same trend of decreasing as you go lower in depth, while polystyrene and PET are still in great quantity as you go deeper in comparison to LDPE.
Since we're still processing the samples, there's a lot that we don't know. But we do know that the weather has impacted the vertical distribution of plastics to some degree. So looking at the data that we have, polyethylene is most prevalent at the surface in higher depths, given its lower density. PET has been found at moderate to lower depths, and polystyrene varies, given its density range, but it is most prevalent at lower depths. And this only accounts for a very small period of time. It's a very small window. And copious plastic particles may have also entered the sediment traps, so we won't be entirely sure until they're brought to the surface for any kind of analysis. But plastics that remain in the corral for longer periods of time are subject to biofouling, so this may greatly impact the distribution of plastics by causing them to sink. And so as we begin to process the more recent samples, we're starting to see this come into fruition. Once plastic debris enters a body of water, it's rarely recovered, so it's going to biofoul over time. And we believe that over the course of the preceding sampling periods, water samples that are collected will contain plastics with similar characteristics as those which degrade and biofoul in lakes and other large bodies of water. So therefore, the new data that is gathered will likely be more applicable, embodying the fate of plastic particles that enter similar bodies of water.
I would like to acknowledge the New York Sea Grant College program, the plastics team, and especially Chelsea Rockman, and the LSAM program for helping fund this project. And I would also love to thank Dr. Hoffman and Dr. Tyler for helping us work through this project all summer. Thank you, Dave Brault, for allowing us to work at the Tate Preserve, and the RIT Plastics and Ecology Lab. Thank you.